When we talk about the natural history of the Earth, we also have to talk about the ways in which scientists date specimens collected so they can put them in a timeline to help us generate a natural history. Now, there are many ways that scientists actually do this, and the important thing to know about this is not so much how these methods work, at least not for the context of a biology class, but to know which are the methods and why scientists can use them reliably. Because the whole thing is that scientists do not look at each of these methods in isolation, but usually they use 2 or 3 different methods to corroborate each other to actually establish the date of a specimen. And that's why we can trust these dating methods, because whenever we use more than one, they end up saying the same thing. So that means that we can use the methods because their accuracy, they're based on physical and chemical principles or biological principles that can be trusted. So the data repeats itself over and over again. Now, of course, there is exceptions, and there's events where mistakes have been made, but these are the minority. And just because there have been some examples of contaminated specimens leading to erroneous results, it doesn't mean that all of the methods are dated wrong. So, what are the methods used by scientists to date things? In this video, I'm not going to get into detail about how these methods actually work and how scientists actually use them. We just have to talk about what are the methods. If you're interested in finding out how these methods actually work, look at the links in the series down below so you can see a little more detail about how they work, but they go beyond the scope of what you need to know for a biology class. Now, one of the most classic ways in which scientists use to date things is look at radiometric dating. And this is basically looking at isotopes which are unstable and decay over long periods of time. For example, carbon-14 can only remain stable for 5,730 years. After that long goes by, half of carbon-14 that you had is going to decay. And it's based on that principle that scientists can date living things, because there's a normal amount of carbon-14 in the atmosphere, because carbon-14 is constantly generated by interaction with solar particles with the nitrogen that's present in the atmosphere. So the amount of carbon-14 in the atmosphere is usually constant. Then that means that some of the plants are going to get that during the photosynthesis, and then animals will get some of that off the plants. But then after the plants and the animals die, they will stop gathering carbon-14, and then whatever they had is what they have. But their carcass, after a while, the carbon-14 that's in it will decay. If you find a carcass and find exactly the same amount of carbon-14 in it as there is in the atmosphere, that means that carcass is just died. But if there's a lot less, you know there's a long time has gone by. And that's the principle behind it. And remember, you don't need to know how it works. You just need to know that we use carbon-14 dating to date specimens. But unfortunately, because the amounts of carbon-14 in the atmosphere are so small, you see it's 0.0000765. After 3 or 4 half-lives, there's such little carbon left in the carcasses that you can't really use this to actually date things anymore. So it's only very reliable to things after like about 20,000 years. Anything past that, you cannot use carbon-14 dating reliably anymore. But that's okay, because there are other kinds of dating that you can use. For example, the radioactive isotopes in the uranium-lead series are constantly used by geologists to date rocks as much as billions of years old. And so this is a process that's used by scientists to date things or to find out where they belong in the timing of their natural history. And that's how we can tell that some rocks are as old as the times of the dinosaurs and so forth. But we also do that in conjunction with other types of dating. For example, scientists also look at sedimentation rates and erosion rates to find out how old rocks are. Because scientists know how long it takes for sedimentary rocks to form, how long it takes for the stratification or for rocks to pile on top of each other and form that rock. And they also know how long it takes for rocks to become eroded. They know how long wind and water and waves will cut through the rocks and make the rocks become eroded. So based on these things, the scientists can figure out how old a piece of rock actually is. This is, of course, based on that idea of uniformitarianism, which is the idea that the Earth is changing in predictable ways over long periods of time. So since it changes in predictable ways, you can use that to date the history of the Earth. And that's how we know the Earth is so old, and how we know sometimes that rocks are millions of years old. So again, in addition to radiometric dating with things like carbon dating and uranium dating, the stratification rates and erosion rates of rocks can tell us how old rocks actually are. But these are not the only things that scientists use. Scientists also use tree rings, because it takes a certain amount of time for trees to grow. And trees will basically make one ring around their, the bark every season. So depending on how many seasons you have in a certain area, you can actually count how many years have gone by counting tree rings. And there are trees which are thousands of years old because they have that many rings. And so we can use that to time the history of the Earth. They can also look, again, as we talked about before, at index fossils, which are fossils that only exist during certain areas, certain periods. And so you can tell how long that a rock actually is. You can also look at sediments and ice cores, which have evidence of passage of time in them. And you can also look at computerized molecular data from DNA, which is the ultimate way to silence the critics of the timing of the natural history, because DNA differences gather over periods of time in predictable rates. Mutations occur at a certain rate. So if you know an animal is different from another animal by a certain percentage, you can tell how long ago this animal split from another. So this is called molecular clocking, and we talk about this in more detail when we do the taxonomy lecture series, so I'm not going to talk about it right now. But molecular clocking, which is based on analyzing and comparing DNA sequences of different organisms on Earth, can be used to determine how long ago these two organisms shared a common ancestor. And since this happens in a predictable biological way, we can actually estimate the age of these specimens. And we can actually figure out that they actually corroborates with fossil evidence, with climatological evidence, with stratification evidence, in erosion rates, and in index fossils. It all corroborates the same thing, that the evolutionary process is indeed happened in the history of the Earth, and that the natural of the history of the Earth, as presented by modern science, is accurately presenting what actually happened in the timeline of the Earth. Alright? So in the next video, we're going to talk about other evidence that's collected by scientists to talk about evolution and the natural history of the Earth. I'll see you guys then.